Okay, so hello again. Excuse me. Okay, hello again, dears. So, dirita ni end, right? So, this is, again, we'll start now with the different types of cultural media according to use. So, according sa yung sa siya paggamit. Like niya. Charot, okay. First is, of course, you have simple, basal, or general. By the definition itself is simple. Basically, it's just plain. He's a plain B. Okay. Basically, ginagamit siya for routine use, and it can support the growth of many organisms, even mga both gram-positive and gram-negative. Okay? And usually, pwede gani fungi makagrow ani. So, wala siya added supplement. It's just plain agar, usually. And a very good example is you have nutrient agar and nutrient broth. Aside from that, you have TSA and TSB. Trypticase soy agar, trypticase soy broth. Okay? You have trypticase soy. Ayan. Trypticase soy agar, trypticase soy broth. Okay. Next, we have maintenance. By the name itself, maintenance. It supports the growth of microorganisms para for future purposes or while in storage. Okay. And a very good example is BHIB. BHIB stands for brain heart. Brain heart. Wala tayo ring light, laban lang kita ni. Okay. Brain heart infusion broth. Okay. BHIB. Okay. Brain heart infusion broth. And NA, nutrient agar slant. Okay. Alright. And next is, of course, you have transport. For transport, these are organisms, or these are culture media used for organisms while in transport. Basically, because there are times ang specimens kay layo from the lab. So, example, na outbreak kunog diarrhea, yes naman, from bais. And then, ang testing kay na pa sa Dumaguete. So, it's quite a long time pa to travel. And, lisod siya if delete nimo siya ibutang culture media while in transport. Example, for COVID-19 specimens, katong i-transfer pa siya to Soto, kailangan dyan siyang specimen transport because pwedeng ma-deteriorate or pwedeng mamatay ang organisms while in transport. So at least manggani, while in transport, they are in storage or they are in culture media para makapaday na dyan po nilang growth and masupply rin yung nutrients. Okay, and a very good example of transport media, you have Carey-Blair medium and Stewart's medium. For Carey-Blair medium, usually ginagamit natin siya for stool specimens. Yes, so katong mga outbreak of diseases, outbreak of diarrhea, mga inana kunuhay. And Stewart's medium is an all-around na siya na transport media. It can even support viruses. Okay, na transport media. Naray pang-add mga additional supplements kung for viruses na siya na transport media. Okay? Alright. And next, you have your enrichment. When we say enrichment, by the name itself, you want to make padaghan, charang konyo. You want to let a specific organism proliferate, meaning padaghanon sa nimo siya before nimo i-culture. Okay? And a very good example is APW. Again, ang sabi na APW, recall, APW is your alkaline peptone water. Okay. Alkaline peptone water, and this is considered to be the enrichment media of choice for Vibrio species. Okay. So sa mga Vibrio species. Okay? So these are, this again, the enrichment media of choice for Vibrio species, APW. So basically, what it does is, ang nutrients na naas APW, it's only specific for Vibrio. So kung naay growth ana mo turbid siya, meaning ang ni-grow ana ra gyud is Vibrio species. And you have other two, selenite F and tetrathionate, usually for gram-neg bacilli, specifically for Salmonella species. Okay? Alright, Salmonella species. Okay. Alright. That's again for enrichment. Please take note. Again, you want to make padaghan. You want to proliferate. You want to let an organism proliferate, a specific organism, before nimo siya i-grow, i-culture sa plates. Okay. Next, of course, differential. By the name itself, differential, you allow the visualization of metabolic differences. So example, ana is BAP and MAC. So you identify bacteria or you differentiate two groups of bacteria or two species ba, two genera, depending on their ability to metabolize something. So example, for McConkey agar, which is your MAC, again, you differentiate bacteria through their ability to ferment lactose. Okay? So again, this is an example of a culture, a McConkey nanay growth. If the bacteria ferments lactose, then you have pink colonies. And if these are non-lactose fermenters, then these are colorless colonies. Again, I mentioned na, kinsa to mga rapid lactose fermenters, di ba? We have Eke, Escherichia, Klebsiella, and Enterobacter. Again, we'll go deeper ani sa biochemical testing. So sa intro to micro, medyo ano to siya, superficial. But when you go to biochemical testing for gram-neg bacilli, mas intense atong discussion ana. Okay? Alright. And a good example, another one is blood agar plate. We differentiate bacteria through their ability to hemolyze blood or through their hemolytic patterns. And we have 3 types, di ba? I'm sure you're finished with Streptococcus species, right? So these are the different types of your hemolytic pattern. First is complete hemolysis. As you can see, your blood agar plate is really red, okay, if walay-walay tubo. But if beta hemolytic ang kagaw, it clears the area. So this is beta hemolytic, complete hemolysis. And that is a good example of ana is Strep pyogenes, right? Streptococcus pyogenes, Streptococcus agalactiae. These are all beta hemolytic streptococci. Okay, and next you have incomplete hemolysis. This is your alpha hemolysis. A very good example of an organism that exhibits this is Streptococcus pneumoniae. Okay, please take note, your no Lancefield antigen, di ba? Streptococcus pneumoniae, incomplete hemolysis, alpha hemolysis. And of course, gamma hemolysis, the medium stays as it is, color red, ragyapon, without any clearing or greenish discoloration. You have gamma hemolysis, usually other strep. Okay? Alpha hemolysis guys, how do you know na alpha hemolysis? Na greenish na discoloration. Okay? Alright, now actually your blood agar plate is one of the primary plates, meaning usually appeal siya sa mga primary plates na itong ginagamit for culture, for specimen or for isolation. Because again, usually other bacteria or most bacteria mo exhibit na silang different characteristics there is a blood agar plate. Okay? But again, in terms of differentiating through hemolytic pattern, we usually use this sa mga Streptococcus species. Okay? Alright. Next, we have your selective. Okay, when we say selective, you want only to grow a specific group of organisms. Example, ganahan kang gram-positive ang mutubo, ganahan ba kang gram-negative ra. So how do we achieve that? We use different antibiotics or inhibiting substances or inhibitors. A very good example, you have MAC. Okay, sir, so MAC differential, selective po siya? Yes, that could be. A culture media can be mixed na types depending on use. Pwedeng selective differential, pwedeng selective enrichment, and so it depends on the composition pa rin and the purpose of the culture media. Okay, so you have MAC, McConkey, BSA, bismuth sulfite, agar, XLD, xylose, lysine, deoxycholate, and MSA, or familiar, mannitol salt agar with 7.5% salt. Again, kanisala guys, nani sa gram-neg bacilli, sa stool culture, don't worry. I'll explain that further there. MSA, mannitol salt agar, 7.5% salt. So it makes sure na ang mutubo ra ana kay Staphylococcus species. Because your Staphylococcus species are very halophilic. They love salt. Maganan sila asgad, char. Okay, alright. 7.5% salt. Now, these are example of some inhibitory substances. So these organisms, these substances inhibit gram-positive. So please take note, crystal violet, basic fuchsin, and as I mentioned, bile salts. Inhibits gram-neg, you have alcohol and chloral hydrate. Potassium tellurite and sodium azide. And of course, if you want to inhibit the swarming of Proteus, you use alcohol and chloral hydrate. Now, swarming, guys, again, nani sa gram-neg bacilli na discussion, don't worry. Swarming is when ang characteristic mo na motility sa Proteus kay mo swarm siya. Like, example plate, then mo culture ka here, ihang motility kay mo ana, na is swarming. Okay, so mo spread siya. Alright? So, basically, if na ay mga case studies, na the organism is a gram-neg bacilli that exhibits swarming, presobuzzer, that is most likely Proteus. Okay? Alright, Proteus. Alright. Next is, of course, enriched. Okay, when we say enriched, these are just from basal or simple. Example, nutrient agar, tryptic soy agar. Butangan natin siya supplements. Example, pwede ba siyang blood, horse blood, sheep blood, para may mong blood agar plate, or chocolate, Cadbury or Toblerone. Joke lang. Lysed blood, again, for chocolate agar. So usually, these are types of culture media to support the growth of fastidious organisms, meaning those that require mga special treatment, mga pa-special, mga imong ubang friends dira. Charot lang, again, fastidious. So example, you have Haemophilus and Neisseria. These are example of fastidious organisms. They need special mga requirements. And we also have the Streptococcus. You have your nutritionally variant strep. Okay? An example, Ana, is you have abiotrophia and granulicatella. Basta na-mention ni Ma'am Teddy, hopefully. Granulicatella, these are nutritionally variant. They require vitamin B6. Okay, sa ilahang vitamin B6, sa ilahang culture media. Abiotrophia and granulicatella. Okay? So, these are your nutritionally variant strep. Okay? So, under Streptococcus family, they need vitamin B6 or pyridoxal. Okay? Pyridoxal. Hopefully na-mention ni Ma'am Teddy. Okay. Alright. So, again, that's for enriched. Usually for fastidious organisms. Okay. Alright. Next is you have special. Sana all special. So, special only for a specific type of organisms. A very good example, you have LJ medium and Middlebrook 7H10. That's for MTB, Mycobacterium tuberculosis. Please take note sa LJ medium. LJ medium, cauliflower colonies. Ayan. Sus ko. Pag kanimo gawas, cauliflower colonies, that's really MTB. Okay? Don't forget MTB. Yeah. And of course, you have your TCBS, thiosulfate citrate bile salt, sucrose agar. That's for Vibrio species. Again, we'll have a different discussion for this sa stool culture na. Okay? Alright. And again, di ba, recall sa MTB, usually we don't opt for culture for diagnosis. Dito rata sa acid-fast staining sa atong slides, right? Because again, dugay ni siya motubo. Alright? So, namatay na ang patient, wala pa na tubo ang kagaw. So, we opt jud for diagnosis to usually use X-ray ba and katong slides, DSSM. Okay? Alright. Lahat itong DSM sa ano ha? Okay, alright. Alright. And of course, you have others. You have anaerobic media. Again, for the anaerobic organisms, those that do not require oxygen, so they have their own list pod of culture media that you need to use. Okay? So, example of that is cooked meat media, again for proteins, and thioglycolate broth. Ayan. Usually, ang thio is not only for anaerobic, pwede po siya makasupport ng aerobic, right? As ma-remember ninyo sa katong mga obligate aerobes, katong na discussion. But when you want to use it for anaerobic media, you want to make sure that this media is free of oxygen, of course. And this media, it has an indicator known as your resazurin. I think na-mention na ako ni. Resazurin, okay? Resazurin turns color pink. It makes the media pink if it has too much oxygen na. So how do you make sure na wala siya oxygen? You boil off thioglycolate broth. Why? To drive off oxygen. Okay? So again, resazurin, it makes the thio broth pink if it has too much oxygen na. Okay? And what do you need to do para mo walang oxygen? You boil the media to drive off oxygen. Okay? Alright. Next is, when we go to anaerobic media, guys, maybe dili na ko, maybe we'll have a discussion on anaerobic culture, but in terms of media, basta di-discuss ko na sa lecture. Okay, it has a different kind of list pag yun of media na not used in routine bacteriology. Okay, because again, daghan kayo siyang requirements because these bacteria are very special po. They don't need oxygen or they don't want oxygen. Okay, all right. Next, you have medium for susceptibility testing. We only have one, and that is your MHA. Always, Mueller-Hinton agar. We'll have a discussion on this. Furthermore, when we go to AST or mga testing for susceptibility. And ang agar depth is always at 4 millimeters and the pH is at 7.2 to 7.4. Now, these are two types of tests for susceptibility testing. We'll discuss kani guys pohon soon na gyud, mga later part na. Okay? You have your Kirby-Bauer disc diffusion and you have your epsilometer test or E-test. As you can see kani mga circle guys, these are known as your zones of inhibition. Zone of inhibition. Ayan. So basically, it's the point where wala na nitubo ang kagaw kay ni-diffuse ang antibiotic, dirira ko ang diffusion, so meaning wala na siya nitubo. And we usually measure the diameter, okay, of these zones. And if unsa man gani iyahang measurement, there is a different interpretation. It could be resistant ba siya, intermediate, susceptible, inana. Okay? Alright. That's for susceptibility testing. And last is of course for biochemical testing. Ayan naman. So sa intro to micro, this is just giran through rin ako ni. But again, when we go to biochemical testing sa different groups of bacteria, we'll go deeper into this. And a very good example is again your TSI, triple sugar iron, right? You have the slant. It determines the ability of the bacteria to ferment lactose and or sucrose. And you have sa butt the ability to ferment glucose, okay? And we have different letters or how to interpret it. If yellow gani, that's always letter A. And if red siya, meaning non-lactose, K. Okay? How do I remember it's on RK? Red K. AY, ayan. Academic year, RK. AY, RK, lang. So, basta yellow gani ferments the sugar, and then red not ferments. So, how do you interpret this? You start first with slant. So, example sa first one, that's A again over A. Okay, so A ang slant, A ang but. Okay, all right. And again, there are different also mga other mga other results. You have gas again if na mga cracks or bubbles or displacement, meaning musaka ning tube media, ang media. You have also H2S production if na blackening. So sa first picture, kaning first reaction, this is A over A with gas. Ano na siya? Because again, na mga cracks. And of course, ang number 2, that's K, red, over A with, or let's say H2S, positive. Okay? Alright. We'll go deeper sa biochemical testing sa gram-neg bacilli. Don't worry, guys. Okay? Yes, I'll do my best. Those are my favorite topics na. Charot, okay? Alright. Ayan. Okay, now for the next part of the video until the end, we'll now go to the different steps in media preparation until the end, the different types of culture media. So yeah, please, let's see each other to the next video na po. All right?